Hiya, this is your first practical video, and the first practical that you'll be doing for your GCSE is looking at onion cells and cheek cells using a microscope. So the first thing that you need to do is be able to use a microscope comfortably. So what you need to know from here is that you look down this is called the eyepiece lens. You can take your goggles off when you look through here. So you look through the eyepiece lens, and then you put your sample on what we call the stage. So you put your slide on the stage, and then you have 3 lenses to choose from. These are objective lenses, and they magnify the image. You always start with the smallest objective lens when you're looking through here, the eyepiece lens, and you get bigger and bigger until you see what you need to be able to see. The wheels on the side, either side, they're called focusing wheels. The coarse focusing wheel will move the stage up or down, allowing you to get a clearer, detailed image. And once you have a clear enough image, use the fine focusing wheel to really get a crystal clear image. So that's how you use a microscope. You also need to know for this practical how to prepare a microscope slide. And the first microscope slide that you have to be able to prepare is an onion slide. And for that, you will need some pieces of onion. You'll need a white tile. You will need a microscope slide. You will need a cover slip, and these are very, very thin. So when you pick them up, just rub them gently between your fingers to make sure you picked up one. These break really easily, so be careful. So the first thing that you need to do is, because you'll be using a stain, and the stain you'll be using here is iodine, you must tie your hair back and you must wear goggles. Goggles do fit over glasses, so don't worry if you wear glasses. Okay? The first thing that you do is you get a piece of onion. You'll be given pieces like this, and you need 1 layer of onion cells. The easiest way to do that is to give it a snap, and a layer should be able to peel off if you pick it up like this. Okay, that's a bit small actually. It may be easier for you to use forceps, so that's what these forceps are here for, to pick it off if you find it easier. Because I've got nails, I'm just going to use my fingers. So you should get a thin onion slice like this, nice and thin, 1 layer, 1 cell thick. You put that on your microscope slide. You're then going to use a dye, and this dye you're going to be using is iodine water, and that allows you to see the cells clearer under the microscope. And you put no more than 2 drops onto your sample. You get your cover slip, and you put this onto the onion slide like this, press it down, and it will hold in place. Then you put your microscope slide onto your microscope, and you observe it like we did before. So each of those the boxes that you see, each of those is 1 onion cell, and depending on how much dye you've used and the magnification that you're at, you could possibly see a dark circle in between each box. That dark circle is the nucleus. The second slide that you need to learn how to make is a cheek cell, and for that you'll need another clean microscope slide. You will need another cover slip, and you will need some cotton swabs. Now you can use cotton swab, a lollipop stick will do the same job, whatever you're given. You take this and you run it on the inside of your mouth quite vigorously, so this will be full of cheek cells. And then what you do with this is that you swipe this onto the microscope slide, and it's really important that you put this in the beaker of sterilising fluid that you'll be given. You must remember to do that because it's got your DNA on there. Once you've put you swiped your cheek cells on there, you're going to use another dye, but this one's called Methylene Blue. Again, you will need no more than 2 drops. 1 drop of this actually will be enough. Onto there, you get your cover slip, put that onto the slide, let that dry off a little bit, and then you put that onto the stage and you observe that as normal. What you'll be requested and required to do is draw what you see in your lab books. Be a teacher will explain more to you about that.